Hello, everyone. I'm delighted to be opening another Global IMMUNOTALK. I'm, as always, connected with the co-organizer of the Global IMMUNOTALKs, Dr. Carla Rodlin, and our fantastic speaker today is Dr. Richard Locksley from University of California, San Francisco. So we are delighted to have him. And so briefly reminding the goals of the Global IMMUNOTALKs, we'll never get tired of reminding this. And these goals are to bring excellence in immunology all across the world in an egalitarian manner and without traveling, making these talks inclusive, and accommodating, and environmentally friendly. And we can do this thanks to the generosity of our fantastic speakers that have shared their knowledge throughout the year. And just a reminder, next week we will be ending the 2020 Global IMMUNOTALKs with Dr. Federica Sallusto. And just a brief announcement, we already have the program for the 2021 Global IMMUNOTALKs, the first half of 2021, and we will be announcing that soon. So with that, Carly will now introduce Richard. Thank you.
Thank you so much, Alina. And of course, we're very excited to welcome the new year with new speakers. So today is my absolute pleasure to introduce our global immuno speaker, Dr. Richard Locksley. Rich is a professor in the departments of medicine and microbiology and immunology at the University of California in San Francisco. And he's also an investigator in the Howard Hughes Medical Institute. And I should also say he's a director of the Sandler Asthma Basic Research Center at UCSF. Now, Rich is originally from Minneapolis, Minnesota. And I guess after growing up in Minnesota, he embarked on a long trip, mostly from east to the west of the US. His first stop was at Harvard, where he received his bachelor's degree in biochemistry. He then obtained his medical degree at the University of Rochester, and after completing his residency in internal medicine at UCSF, he came a little bit more to the center and he trained in infectious diseases at the University of Washington. He finally returned to UCSF, where he served as the chief of the Division of Infectious Diseases before moving to direct the Sandler Center since 2004. Now, looking into his CV, I believe, Richard, maybe you can correct me, you have been at UCSF for more than 34 years, which I wonder whether that is also the reason that contributed to you to become an avid fan of the San Francisco baseball team, the Giants. Which I guess is not a bad choice considering that they are the most successful professional baseball team. So I think Rich has been very happy there in San Francisco. Now, more seriously, Rich has been a true pioneer in the understanding of how cytokines regulate both innate and adaptive immunity, with a focus on type 2 immunity. He has inspired many of us with his fundamental discoveries on the biology of IL-4 and other type 2 cytokines, and has developed outstanding and elegant genetics tools to study these cytokines. And more recently, his laboratory led the way in understanding these other interesting cell types known as ILC2s and TAF cells in both allergic and anti-helminth immunity. And I think we're going to learn much more about these cells today in his talk entitled ILC2s, Innate Allergy and Tissue Resilience. Now, for his monumental contributions to the advancement of science, Rich has received numerous awards, and he's an elected member of the American Academy of Arts and Sciences and the National Academy of Sciences in the United States. So Rich, thank you so much for accepting our invitation. We are delighted to have you here.
So great to be here, and thank you for the kind introduction, Carla and Elena. It's so terrific that you guys do this. We're just entering lockdown in San Francisco as cases kind of surge, and these kinds of things are respites for a hopeful near future when we can all get back together and rejoice in being more front to front, face to face, and doing what we love to do. So they're hard times, but these things are so important for keeping missions of science going forward and keeping a human face on it. So I really appreciate the opportunity, and what you guys have done to organize this has just been terrific.
Yeah, thank you so much for those words, Rich. These are very difficult times for all of us, and it's nice to connect, at least in this way, on Wednesdays and also later on through YouTube. Okay, now, one of the things that we hear people really enjoy about these talks, apart from the phenomenal talk that I'm sure we're up for, is to get to know a little bit more about our speakers. Now, you have had a very long and outstanding scientific career and have made truly fundamental and impactful discoveries, which is probably a reflection that you have very good taste on identifying important questions. So what we would love to learn from you today is, what are the areas of immunology that you currently find most challenging and exciting?
Well, thanks, Carla. It's kind of interesting in thinking back to successes. I think some of that is driven by how many years I've been doing this and the terrific people, for sure, that I've been able to persuade to come to San Francisco. When I started really doing research in college days back at Harvard, I was working at Peterman Brigham in a cardiac physiology lab, and all of the intent was on purifying molecules, pumps and muscle contractile proteins, and trying to understand how they work, with fundamental endpoint being to understand how the organ worked. That is, how does the heart contract, and how does it maintain its electrical excitability to be a functional organ. And all of the research was directed at how the organ works. And then shortly after that, when I started my scientific career, the molecular structural evolution revolution really began, and everything went down to the angstrom level and to cloned molecules, and remarkable understanding about how biology works. And then the last few years, we've started to back out and try to understand from these small molecular machines, how do you build a heart? How do you build a brain? How do you build an intestine? How do these cells integrate to maintain their function from the molecules that we now understand increasingly well? And I think that's the next great challenge. And the fantastic thing about immunology is, lo and behold, immune cells play a part in all of these very fundamental tissue-specific physiologic events that we never really appreciated as kind of a ground state of the immune system. So these are cells that get into every organ and do these remarkable things that labs all over the world are discovering every day. And we're using therapeutically in new ways we never imagined. So I think it's a great time for immunology and for young people to think about this as a discipline that just has a remarkable future ahead of it.
Thank you. That's great. So I guess in that note, we will probably learn more about your thoughts and findings on tissue resilience. Let's see if you can share your screen. Okay. Let's see if you put it in presenter. Yeah. That's great, Rich. Looks great.
Okay. So thanks again for this terrific opportunity to talk to all of you out there, and good morning, afternoon, evening. I know people are all over the place, and appreciate your keeping the spirit of this whole thing going. I guess I have to advance.
Yeah, you have to use the lower left. Yeah. Got it.
Yeah. So as Carla and Elena pointed out, our lab has been interested for a long time in type 2 immune disease, or immune disease regulated by Th2 cells, really, in IgE. And this affects enormous amounts of people around the globe. In developed countries, but increasingly now around the world, this presents with allergic diseases. I can let you read the astounding numbers there. The increases that have come in the last few years have move this clearly, or put a emphasis on environmental interactions with evolutionary genetic selection that's unable to be as nimble as the environment has, the speed with which environment has changed. Such that now in the US, over 10% of kids have food allergy of some kind, which you would think of as absolutely anathema for our evolutionary success. And of course, in developing countries, there's an enormous burden of parasitic helminths. I would point out, though, that these are widespread. They're essentially in all feral vertebrates. I think of Barbara Rehrman's wild mice, each having about 3.5 worms of different species per animal, that we live with our entire lives. These animals essentially senesce in our bodies. So type 2 immunity doesn't really end their lifespan. In many ways, I think these organisms have evolved as evolution works to select the sweet spot where we can get by and they can get by. And most of the infection is asymptomatic, largely post-reproductive, and most feral animals are really really infected. Let me just get this pointer right here.
Rich, I think if you select laser pointer.
Yeah, it's on.
Yeah, there. Got it.
There we go. So I want to stop a minute and think back, as you do with these talks, about what really made the difference in getting there. And I think it was development of this rather simple model of Nippostrongylus brasiliensis, a normal hookworm of rats, that is adapted to mice, but then expelled with a very strong type 2 immune response. And of course, we most labs raise these organisms as larvae in rat poop and purify them and infect animals subcutaneously. They migrate to the lung. They burst through the alveoli and get swallowed, just like hookworms in humans. They get to the jejunum, and males and females they have sexual stages and begin laying eggs at about 5 to 6 days in these are days in our facility. And by day 9, 10, they're expelled by a process that is CD4 T cell dependent, 413 dependent, 4 receptor alpha, the shared subunit of 4 and IL-13 receptor, and STAT6, the downstream signaling transducer. And I really want to point out here that Joe Urban, a fantastic agricultural scientist at FDA, and Fred Finkelman, who's in Bill Paul's lab at the time, trying to look for good systems to study IgE switching, really delved into this model and made it part of the everyday armamentarium. And I was fortunate enough to work with Fred and Joe years ago in a summer molecular parasitology course back at Woods Hole for several summers that were terrific. And where I learned a lot about this model and brought it back into the lab because I realized how powerful this was to look at type 2 immunity. And I think you'll see as we go through what we've learned, how many laboratories have used this worldwide. Now I realize across the Atlantic, I'm not slighting the Richard Grensises or the Rick Maisels who spent years developing these models as well. But for me, Joe and Fred were just fantastic at teaching me a lot about the insights of this model. Now, years ago, there were many experiments began to appear, including from Manfred Kopf and others, as people were knocking out IL-4 and discovering what IL-13 did. The McKinsey lab was very informative there. That there was some peculiar aspects of IL-4 minus mice, or when you transferred CD4 T cells that were IL-4 deficient into rag mice, they would reconstitute a lot of the immune response that left just this difficulty in understanding who is doing the heavy lifting, producing the 4, we later found out the 13, as shown really by Fred Finkelman first, that was very important in getting rid of these worms. So David Veringer, back when he was a postdoc in the lab, and Ong Liang is still in the lab, just doing terrific things for us in engineering mice, in the pre-CRISPR era, recombined the IL-4, IL-13 locus to introduce LOXP sites. So we could cross these to various strains of mice, here CD4 Cree to knock 413 out of T cells. And that was accomplished. They clearly didn't make 413, they being T cells. And there was absolutely no IgE. We know almost all of that is due to IL-4 in the mouse. But the remarkable thing is the worms almost came out completely normally in these mice. And we could see by then we had various reporters for 4 and 13 that there were CD4 minus cells in the gut that were expressing these cytokines that were involved, presumably because the 413 total knockout mice clearly were unable to expel these worms. So that was, for us, a very insightful experiment and drove us further into characterizing these cells. And I'm now skipping forward to 2010 when Shigeo Kiyasu's lab in Japan and McKinsey and later ourselves all described these innate ILC2s. I think our contribution was more their distribution across tissues. I will point out that all of these original contributions used the Nippostrongylus model, just again pointing out how there's kind of unseen little moments in biology where things get pushed to the forefront by individuals that you don't think about sometimes. So why did I think that's so important? I think up until that time, when we think about allergy and asthma, we're thinking about this end of the spectrum, Th2 cells that have this pesky little antigen receptor. It is terrific for recognition of allergic disease and spurring memory responses by tissue-resident cells, as we now know. And these cells get to be problematic, of course, and they recognize antigens from food or pollens or non-innocuous sources, as happens in allergy. But all of a sudden, we were exposed to an innate T cell without a receptor that was making these cytokines in tissues under non-allergic conditions, where it still made IL-5 that was important with IL-13 and attracting IL-5 into eosinophils into tissue, as Mark Rothenberg and others had shown, and leading to this alternative activated state of macrophages. So this gave an insight into perhaps using these cells, you could begin to understand what was the foundation of this type of immune response in the basic physiologic unit of health. How does the body work, and how does this get conscripted by the type of responses that go haywire in the pathology we think about with allergic disease? So Steve Van Dyken and Jesse Nussbaum, when they were in the lab with the help of a number of others, went through pulling these cells out. By now we had pretty good reporters to mark IL-5 and 13 in these cells and compare them to tissue Th2 cells that express these cytokines to show that these cells had a common transcriptional landscape as mapped by RNA-seq. We got help from Jimmy E. and Alex Marson at UCSF at that time, that they shared chromatin accessibility by ATAC-seq. They shared a super enhancer landscape across critical cytokine effector domains. And most interestingly, I think that the terminal differentiation of these cells occurred as they entered tissue. And we could see this because the reporter alleles blinked on only when these cells moved into tissues and got appropriate localizing signals. All of a sudden, they would turn on this program and be identifiable as these lineages that we call ILC2s and Th2s. And we weren't the only people doing this by any means. Other labs, DeSanto lab, Marco Colonna, a number of others, had begun to look at this landscape, and David Artis, I know, is another lab that did this, in both human and mouse, and show that these share many, many commonalities by organ. And now, the next step in trying to understand this was understanding the ontogeny of ILC2s. Jen Bando, when she was a student in the lab, had realized that precursors of these cells appeared in fetal liver and during embryogenesis about E16.5 or so, 16, and that these cells could be recognized by the expression of arginase 1. So we built a tamoxifen-inducible Cree that would allow fate mapping by mapping with a ROSA identifier allele, and also used Jim DeSanto's ID2 mapping mice as an independent strategy. And we here as Christoph Schneider, a postdoc, and Jinwoo Lee, an MD PhD a graduate student in the lab, is now down at Stanford completing his training. We could label cells in fetal development in neonatal period shortly after birth, or in adult life with tamoxifen, and begin to look at with kind of pulse trace what happens to these cells over life as they're diluted out by unlabeled cells. And what this disclosed was that cells were indeed entered tissue and developed in fetal life, but that the biggest proportion of cells occurred shortly after birth, the first few weeks after birth, in this neonatal perinatal period, when cells expanded quite dramatically to fill up tissues with cells that could be followed over up to 2 years in the mouse. And the fetal cells slowly disappeared from organs, and the neonatal cells as well. And there was a replacement by adult cells that were being made later in life, as we could show by labeling in this period, that varied according to the turnover of the tissues, such that tissues that turned over rapidly, like bone marrow or skin or gut, the turnover of cells and replenishment by adult-derived cells was more rapid than organs that turned over more slowly, like lung and fat, where turnover of these cells was quite slow. So with this in mind, we could begin to compare what happened during this seeding period, either in the neonatal window or of these adult, more stable cells. And when Jinwoo and Christoph did this, it was clear that cells taken from fetal or adult tissues segregated with the tissues from which they came. That is, skin cells from the neonatal period, well, ILC2s we're looking at here, or adult cells segregated with skin ILC2s from the target organ sorted with where these cells transcriptionally resided. And the same is true of lung and small intestine and all organs that we looked at. But in all cases, the neonatal cells had a more activated phenotype if we looked honed in on their effector cytokine potential. So here the fetal cells in this perinatal period are really auditioning all of the effector function that we think about of Th2 cells, including IL-13 and 5, GM-CSF, AREG, CXCL2, along with what we knew at the time, this inflammatory phenotype with the IL-17A locus, as these cells, as shown by the ATAC-seq look that we did, it maintained a poised ROR gamma T locus in tissue. So always, if we looked at these neonatal cells, there was more IL-5 here as compared to the adult cells. Same with IL-13. You could also see it in the lung where the fetal cells are blue here, had higher expression of IL-5 and 13. And the intriguing thing is, although we didn't have all those reporters into germ-free mice, which we're doing, is we could take these cells out and use qPCR to really show that the numbers of ILC2s and their transcriptional profiles really looked exactly the same in germ-free mice, suggesting that unlike ILC3s, sets of cells that are driven by AHR ligands and very much linked to the microbiota and to the diet, these cells seem to be on a developmental trajectory that seems in large part to be independent of the microbiota. This doesn't say the microbiota don't have an impact on the ILC2 number or how they develop. It just says that they're not required for this developmental trajectory. Now, we move into adult life and look at bone marrow cells as compared to peripheral cells. We could see that there was quite a bit of heterogeneity, not only among these individual tissues, but within a given tissue. Here in small bowel, you can see that there are ICOS high and low, or in IL-2R beta across ILC2s in these tissues. But there's something I want to emphasize here that I'll come back to. These cells were all selected for expression of IL-5. We knew those are ILC2s, as I'll get into, nomenclature that was set by august committees. These cells are selected by arginase 1. We knew from Jin Bando's studies and from fate mapping that these cells constituted the ILC2 precursors in bone marrow. There's no effector cytokines in these cells, so there's no, they don't express IL-13, for instance. They're very low for Array. But what you can see is there's quite a bit of heterogeneity in bone marrow. Some of these cells are expressing TBET, some RORC. These cells are all innate lymphoid precursors, but there's quite a bit of heterogeneity. If you look even more carefully, all these cells are GATA3 positive, 7 receptor positive, but you can already notice there's GATA3 low, GATA3 high, cells that express 18R receptor that we'll get back to, cells that are negative, and cells that are high for 25, some co-expressing cells here, and 33. And what's interesting is that the mature cells, the IL-5 expressing ILCs in tissue, have quite different alarmin cytokine receptors in tissue, like IL-18 in skin, 25 in small intestine, 33 in adipose and lung, that was independent of these, the necessity of these alleles. That is, if we knocked out the ligands for these receptors, they still develop this way, as if there's an anticipatory nature of the positioning of these cells and organs in order to see certain types of perturbations later in life. And understanding that gets back to these questions I raised with Carla in the introductions about how do tissues organize into functional states and what role do immune cells play. So what's in a name? So I have to pause here a bit and acknowledge a lot of other people have contributed many things in this field. And all of these things were happening very, very rapidly around 2010 to 2012. And Ergen Spitz really organized a group of people who are involved in the field, a number of labs all over the globe, and ended up with this nomenclature. I had always thought we should just call these IH1, IH2, IH17 in honor of DNAX, where these were the TH cells are originally, the nomenclature was found, and some of the early discoveries of IH2s were really made. But the committee really came to this ILCs, which I think has worked well, and that they be named by the cytokines and the transcription factors that regulate their function. And that was based on their similarity to TH subsets. And that was reinforced in the revisiting of this recently, 5 years later, in 2018. What I want to emphasize is that by doing that, of course, we don't see with our mice, we don't see precursor cells, we don't see the trajectory of cells who might turn off these reporters. And so I want to summarize a lot of data, many of it tangential to things done in our lab, some of which we contributed small bits to, but I think are important in understanding this study. And I've cribbed a cartoon from a superb paper by George Geistiger's group, really looking at these cells in the lung. But I would call your attention to a number of contributors around the world, starting with Diefenbach, and I particularly want to shout out to this Piotr Bielecki and Sam Riesenfeld's study in bioarchive that's under revision now from the Regavin Favel labs we involved a little bit with, that have looked at this in skin with quite amount of detail, with a lot of similar findings, but also the McKinsey lab, Bendelac, the Bandula, and a group looking at Takai group, looking at IL-18R1 precursors. So I'm just going to sketch these out based on this cartoon that synthesized a lot of these data from George's nice study in immunity that's published now. These rely on, describe this precursor that's been found in both humans and in mice. PD-1 high is originally shown, I believe, by McKenzie's group. TCF7, PLZF, as Bindelac so carefully has delineated, and TCF7 that move into bone marrow, move into blood as circulating ILC precursors. There's a subset of these that expresses 18R receptor and 25 receptor that is, I believe, enough data is mounting that is a precursor for ILC2s, already perhaps pre-committed. These cells move into resident ILCPs, probably ILC precursors in all tissues, but the ones, again, probably best looked at are constitute the skin, lung, and small intestine. And under homeostasis, ILC2s develop, turn on these reporters that we've been following, IL-5, 13, and under, when pushed and activating ROR gamma T, the 17A receptor. And under homeostasis, you see these in tissues undergoing basal interaction as they promote aspects of tissue health that we can talk about. When perturbed, here again with in brasiliensis, kind of this kind of star way to perturb this system because it impacts so many systems, these precursors are pushed in a transition from IL-18R high, TCF7 high, to GATA3 high, ST2, that's the IL-33 receptor, through a proliferating transit amplifying stage, you might think of these, to a plethora of effector types, some that might have regulatory roles expressing CPRG, some that are more Th2-like with IL-13, others that may expressly talk more with T cells or Treg cells, others with different kind of phenotypes. I think in the Regev lab, a little bit different analysis was done, suggesting these cells are perhaps in much more fluid states than in discrete entities with a different type of analysis, but arriving at many similar kind of findings. In that lab, we could use fate mapping of our mice to demonstrate using the Cree as a primer in the RNA-seq, precisely that these cells can give rise to many of these cells directly, although precursor cells can directly contribute to each of these compartments without going through intermediate stages. So where do these cells go? So this is an important finding by Tony Wong when he was in the late Bill Paul's lab, describing these inflammatory ILCs in blood. These are IL-25 responsive, lineage negative KLRG high cells. I didn't point it out on that earlier RNA-seq. We already knew this is the phenotype of small intestinal resident ILC2s, a high microbe, high nutrient organ where these cells reside normally. Then in a beautiful paper where Tony collaborated with Ron Germain to look, again with Joe Urban providing some heavy lifting in both of these in the Nippo model, to look at how these cells moved from the gut by upregulating S1P receptors, much like lymphocytes trafficking out of lymph nodes, to enter lymphatics and then the blood, and finally could show up in lung to contribute cytokines that impacted the status of the health of the lung in this by moving into blood. And here the Grensis lab showed that you could use a entirely mucosal parasite, that is one that never goes through the lung. And these cells are still subject to exuberant proliferation. The terminal effectors move into blood and then can move and to systematize the response. Now we looked at this a bit by using our fate mapping mice to try to understand it a little bit better. Here are the number of KLRG5, RED5 ILC2s In lung, they're at the limit of detection. In blood, I'm sorry. They're at the limit of detection. There's a small little blip at day 2. We can talk about if you're interested afterwards. And then between day 4 and 5, these enter blood at very high numbers that others had described. And this drops down at about day 14 and is essentially back to normal by day 17. And if we characterize receptors on these cells, there's very few in uninfected mice. On day 5, most of these cells express the IL-25 receptor, here characterized by IL-17RB. And by day 13, most are low for this receptor and express ST2, the IL-33 receptor. Now, by using this fate mapping mice, we could treat mice, again, this is Christoph and Roberto and Jin Wu, treating mice with arginase 1, waiting for a month after they've been pulsed and letting the signal be diluted out depending on turnover of ILC2s in individual tissues. And in this way, bone marrow largely dilutes the signal out. Small intestine is diluted down to about 10 or 12% of cells are labeled. And lung, which turns over slowly, is still quite highly labeled. So they adapt the turnover rate of the tissue in which they reside. And if we look at a course of Nippostrongylus, this stays true. That is, the cells, the ILCs in the bowel are still labeled at about 15% or so, and the ILCs in the lung are diluted out a little bit by the inflammatory response in the lung, but they're labeled at about 50% or so. And now we can look in the blood and see that the cells in the blood at day 5 are largely coming from bowel, and day 13, even though they go through the lung first, are coming from lung, or at least have the same label as cells that come from that tissue, a label that's put in long before the experiment. And you can also see this just by genetically. If you knock out IL-25, the label for that is constitutive for the IL-25 receptor, the ligand for the IL-25 receptor in small intestinal ILCs, you lose the day 5 blood wave, but not the day 12 wave. Whereas if you knock out IL-33, the receptor constitutively expressed on most lung ILC2s, the day 5 wave is intact and the day 12 wave is lost. So then Christoph did a very clever experiment, that is treating mice with this migratory L3. They go to the lung, and then treating animals with a drug that kills the worms before they get to the gut. And when you do that, you lose that first wave, but not the second wave that comes from lung. Conversely, if he takes L5 larvae that now are able to live in the lung, purified from a rat lung, and gavage them into the guts of mice where they start to lay eggs, the first wave is lost. They've bypassed the first wave is left intact on day 5, but the second wave is lost. So even though they get to the bowel second, the wave there comes first. And you can see that if you go directly to bowel, you don't get the expansion in the lung by these local ILC2s. So what this tells you is, we'll talk about this receptor-driven program in bowel is quite adept and quite rapid as compared to what we think of as an IL-33 damage-driven niche exclusion driven by damage and IL-33 release from nuclear localizations. So I'm going to summarize a bunch of data from largely from other laboratories, but the way that we think about it, the way I think about it anyway now, is that most tissues are endowed with a precursor pool put down during fetal development and under constant slow replenishment. Cells under homeostatic conditions are under states under IL-5 expressing conditions that we call ILC2s, or activated conditions where they express 13 that are tied into the homeostatic needs of that tissue. And I'm providing an example here. Jesse Nussbaum did some years ago, small intestinal ILC2s, here labeled with red 5, but here with both IL-5 reporter and 13 reporter mice. If we fast these mice, small intestinal lamina propria ILC2s relatively quiescent for cytokine expression. When you gavage these mice with nutrients, these cells turn on IL-5, MFI goes up, and they turn on IL-13. So many of these cycles are going on in all tissues tied into the physiology of that day-to-day cycle. We know now from Geistiger's work and Regev and Pavel that there are a number of states and others of these cells, David Artis, Vijay Kutru, but then under strong perturbation, these cells can be pushed to turning on RO gamma T dependent cytokines 17, IL-22, in some cases turning off 5 and 13 and becoming X ILC2s. And these cells are the cells that get extruded into the circulation, where either the cells directly or the cytokines they make, which you can now measure in the blood, work back at mucosal sites to reinforce mucosal resilience at distant sites, as shown by Richard Grinsis, for instance, and the Tony Wong studies I mentioned. And of course, precursors are now re-entering to fill this empty space as these cells move, as Sam Reisenfeld showed in the Regev lab, can move directly from the precursor stage or from all of these intermediate steps very rapidly to converge on the need for rapid response to respond to perturbation. And I think understanding what happens to these cells, whether the mature cells really re-enter and contribute, whether these cells die, what is the stimulus to drive this precursor pool are major things that we don't understand. So now I'm going to move a little bit into understanding some of these circuits in tissues guided by some of our findings. For instance, the high expression of IL-25 receptor on basal ILC2s in small intestinal tissue. And I'm going to turn here to studies really driven by two terrific former postdocs. Jakob has now got his own lab up in University of Washington, and Christoph, who's at ETH, both doing great things. And absolutely fundamental to all of these studies and all of our studies, Anger Liang, who created reagents that are really allow these things to go forward and contributes in so many ways to the lab. So Jakob really set out to try to find where is the IL-25. We knew genetically there was an effect on some of phenotypes we were looking at, but we couldn't find it. And when he labeled it, it was a little disappointing at first. There was a few, clearly what had to be secretory cells, rare group of cells in the scattered in the enterocyte groups of a small intestinal roll here, blown up so that you can see where these cells are. But when he gave them this terrific model, Nippostrongylus, all of a sudden you can see this fundamental reorganization of the biology of the small bowel. Now, this was rather remarkable and led to really our uncovering the source of IL-25 as tough cells, these rather enigmatic mucosal epithelial cells, and they're linked to type 2 immunity. These are post-mitotic epithelial cells in all vertebrates. They're rare, but present in all mucosal tissues and some unusual tissues like thymic epithelium. They express unusual transcriptome and proteins, the taste receptor signaling cascade. They synthesize acetylcholine, which is interesting among epithelial cells. Prostaglandin D2 and cystineoleucotriene biosynthetic pathways are completely intact, and they make these substances. And their lineage is dependent on the transcription factor POU2F3, an OCT transcription factor involved in their lineage fate. As best we can find, they're really the only source of IL-25 in the mice using our reporter alleles, and that's not to say they are the only one, but within the constraints of the reporter, that's the best we can see. And this just shows with microscopy how these cells from the mouse trachea can reach out into the lumen. And with this, Yaka really genetically defined the whole epistasis. We published this simultaneously with Philippe Jay's lab with many of the same things, using, again, Nippostrongylus to illustrate the power of this system. And what Jakob and Philippe's lab elucidated was the constitutive production of IL-25 from these cells that activate ILC2s to release IL-13 that affects via the IL-4 receptor alpha-13RA1 on these progenitor cells, the secretory state of the gut, such that when a worm is put down the hatch, this system is pushed forward in a feed-forward way through signals we didn't understand at the time to make this IL-25 very visible to ILC2s, which expanded, made 13, drove amplification of this system in this feed-forward loop. And Jakob has gone on to describe production of cystineoleucotrienes, very critical and synergizing during helminth infection to drive activation of the system, although it's still not clear what the upstream components of this system are. At the same time, Christoph became interested in what is the brake on this feed-forward loop, what shuts this down, and was intrigued by studies from Sarah Gaffin showing that A20, the ubiquitin modifying enzyme, modified TRAF adapters in the 17RB signaling chain of the 17E, the IL-25 receptor, to restrain signaling and regulate it by controlling signaling through the 25 receptor. So we collaborated with Avram Ma, who had a floxed allele. Here we're looking at IL-25, at the ILC2s or IL-25 expressing tough cells in resting mice. And when Christoph knocked out, made this IL-5 dependent knockout of A20, we got this wonderful phenotype where animals looked like they had worms, even when they didn't. And remarkably, even though A20 is highly expressed in ILC2s all over the body, there was remarkable little effect in other organs, attesting to the effect, the committed effect of IL-25 to small intestinal homeostasis. And indeed, these animals, despite this large change in the secretory aspect of the epithelium, maintained metabolic state, in fact, a favorable metabolic state in other studies we had done. And they do this by lengthening the bowel, or in association with lengthening the bowel, quite striking, about 15 centimeters or so. And it's absolutely dependent on tough cells. If you knock out IL-25, IL-4 receptor alpha, including on the epithelium, or tough cells, by knocking out, but in the absence of this transcription factor, POU2F3, this lengthening does not occur, and it requires signaling through tough cells through a TRPM5, this depolarizing sodium channel expressed in these types of cells. So at the time, Claire O'Leary was going through different dietary domains as part of her projects in the lab, and we noticed that we had quite a hierarchy of tough cell frequency that was confounding results. If we floxed for receptor alpha, part of the 13 receptor off the epithelium, we got this low stochastic number of tough cells. It was very close to what we saw in either germ-free mice or newly purchased JAX mice. But in many of our strains that were housed for a long time at UCSF, we had significantly more tough cells. And this looked like some of our A20 flox mice, but not all of our mice. Some of the flox mice seemed to have the numbers that we saw in animals challenged with another helminth, Helicmos samoides or Nippostrongylus. So Claire kind of set out trying to figure out what was going on, looking at these mice. Amazing what you see when you open these up. And of course, this is associated with the presence or absence of tritrichomonads, where you can convert a JAX mouse to a UCSF mouse by colonizing the cecum and distal small bowel, or an A20 flox mouse to what looks like a helminth mouse. So what are these tritrichomonads? These are protists, anaerobic protists that were really made famous or brought to everybody's attention by Mike Howlett when he was postdocing Wendy Garrett's lab back at Harvard, when they noticed a number of imported mice in their facility had abnormal numbers of tough cells and giving some of their spurious results. These are evolutionarily ancient organisms that have very primitive mitochondria, primitive ancient evolutionary mitochondria called hydrogenosomes. These cells lack the electron transport chain that we use to very efficiently generate ATPs at the end of the Krebs cycle. In hydrogenosomes under anaerobic fermentation, this reflects the lifestyle of these organisms in their ability to degrade very recalcitrant polysaccharides. These are the type of organisms that degrade cellulose in the guts of termites that couldn't otherwise live without them. And they use a terminal reductase, an iron reductase, to generate acetyl-CoA from pyruvate, and then a succinate transferase cycle to grab CoA and generate ATP. And that leaves short-chain fatty acids and succinate along with hydrogen as the end products that Christoph could actually measure, Christoph and Clara could actually measure in the lumen of infested animals. And the striking thing was that tough cells expressed receptors, both protein and RNA, for one of the free fatty acid receptors, FR3, and GPCR91, suckin RM1. ILC2s don't see succinate. It's really just the tough cells. And indeed, when Christoph put succinate down into animals, these animals generated a response to the ILC2s fired to succinate, but not to acetate. And it was absolutely dependent on the presence of tough cells, IL-25, and the TRPM5 signaling, a little residual signaling through TRPM4 that we know is in these cells. So this is that cycle. These cells, these organisms degrade recalcitrant fibers, generate acetate, succinate, which activates the secretory cycle by activating ILC2s and contributing 13, and now is used, as Jakob showed, on organoids to generate tough cells and for study in organoid cultures. And other people made these contributions at the same time we did, including Jakob in his independent lab, and the Morel Institute using bacteria that generated large amounts of succinate. But the remarkable thing is the adaptive remodeling, metabolic homeostasis, and concomitant immunity, whereby this status of the bowel renders these cells recalcitrant to infection with larval forms. So this adult animal can live by itself and complete its life cycle in a host whose metabolism and adaptive remodeling has enabled it to withstand this infestation. So there are a lot of tough cells everywhere. I don't have time to go through a lot of these things. I'll mention briefly just two brief stories. Clara has been working on gallbladder tough cells. There are a lot of tough cells in gallbladder, and looking at these, you can see a lot of them. They're high for IL-25. These cells do not turn over. They're not expelled at the end of the villus the way happens in the small intestine, so they're quite long-lived. There are a lot of ILC2s. There are also some ILC3s in gallbladder, but these ILC2s express IL-33, and we've been unable to see clear dialogue between ILC2s and tough cells. But tough cells are clearly linked to the physiology of this organ. These cells are regulated by a bile in ways we don't entirely understand. If Claire sequesters bile, which leads to loss of FXR expression and loss of inhibition of biosynthesis of bile, you synthesize bile, you begin to lose tough cells. If you block this with an agonist of this hormone receptor FXR, GW4064, you relieve this inhibition, you drive up cholesterol and bile acid synthesis, and you inhibit bile acid synthesis, and you retain tough cells. And if you take the most prevalent bile acid, cholic acid, including in germ-free mice, and just include it in the diet, you can impact numbers of TEF cells. So Claire's embarking on really a serious look on specific transcripts and proteins in gallbladder as compared to small intestinal TEF cells to begin to get a handle both on this cycle and on the phenotype that she sees in mice that lack TEF cells in gallbladder. So I want to just finish up with what about an organ that doesn't have a TEF cells, and that's skin. This is just a diagram of how rapidly and remarkable the skin turns over from the Fuchs lab that has contributed so much of our understanding. The skin undergoes these cyclical growth and breakdown of the old hair follicle and move to new hair follicles as you generate new stem cell compartments as stem cells migrate to generate the new hair follicle contiguous with this piloerector system and the sebaceous gland. So a remarkable growth system. And Roberto, MD, PhD dermatologist in the lab here, he's looking very handsome as he heads off to dermatology clinic. He doesn't always look that handsome. He's been up in the lab for a long time. He always looks handsome, he just doesn't always wear a tie. Roberto began to look at ILC2s and how they work in a non-TEF cell tissue. And here you can see these cells activate. They're IL-5 positive. They're in hair follicles in the epidermis. At birth, they turn on 13, and now they phase with anaphase, that activating time of growth in the hair follicle. They turn on IL-13. And with telophase, as you turn this off, telogen, you turn this off and IL-13 goes down. There's only a couple of symmetric hair cycles before hair cycles begin to get asymmetric. And so we really can't follow it. But it's not only IL-13, it's IL-22 that goes up in this anagen phase. We know from single cell sequencing studies that these are the same cell, which was interesting. And we know that IL-2 has effects on holding back work of others, holding back differentiation of keratinocytes. And Roberto wondered what IL-13 actually did, that maybe this involved in some interesting biology. And in fact, here he dipilated cells to synchronize hair growth, and this adds a little inflammatory component, but I think it's a fair look across all the animals. And when he adds IL-13 or IL-4 complexes along with 13, you can see this slows the hair growth. And when he pulls out the hair follicular stem cells and looks by EDU incorporation, the proliferation of stem cells is slowed. And when he looks with help from the Spitzer lab at CyTOF, proliferation is largely by Ki67 shut down across a number of cells across the epithelial unit, the hair follicle unit, infundibulum down to stem cells, and STAT6 signaling is evident in all cells in that unit. And so Roberto began to think, well, this is a system that may affect both HFC proliferation and keratinocyte differentiation. This might be really interesting biology. And with that, we have great mess for this. And Roberto looked at our IL-2 deficient mice, our deficient mice, STAT6 mice not here, our 4 receptor alpha mice. And he came back to him and said, all these mice have messed up skin and messed up hair. What's going on? And of course, we had lost a lot of great postdocs. I told you about Christoph and Jakob, and they'd taken a lot of young mice to reconstitute colonies and to get things going. And it's not all on them, but we had a lot of old mice accruing around as new postdocs started up. At the time, the Nagao lab had noted that the absence of ILC2s, sebum changed, and there was changes in microbiota, effects on sebum growth, on sebaceous gland growth, and maybe this was a bacterial phenotype. So sure enough, IL-4 receptor alpha mice re-derived at all of our postdocs labs are perfectly fine. Our mice kind of had a phenotype you could see. We got unaffected mice from the Ensel lab at UCSF, and then Roberto co-housed these mice or left them unco-housed for 3 months to try to see what happens. And it's quite striking. If you co-house a mouse that looks like a wild-type mouse without 4 receptor alpha with a affected mouse, it becomes quite affected. And this is associated with activation of skin ILC2s to the level that we see in our colony at UCSF, extrusion of these cells into blood. I'm not showing you this, but they have IL-13 and IL-22 in blood, and an impact on distal organs as the lung ILC2 compartment begins to take on an IL-18 high receptor phenotype that looks more like skin ILC2s than normal lung ILC2s. Now, the skin also changed when he looked at the hair follicle density. This also changed in hyperkeratinization, but this really exposed the problem. He could look carefully. These little organisms are now quite evident in almost every hair follicle in our colony. This is chitin, a molecule that we're familiar with. And when Anger and Roberto developed a chitin synthase gene tag to find these mites. This is the chitinous structure of these mites. It was clear that these animals are highly infected with mites when they're co-housed, but they're clean without it. So these Demodex mice are these cigar-shaped mites that colonize mammalian hair follicles and xylosebaceous glands. They're in all of us. I know that sounds gross, but they are. It's essentially 100% colonization with age, particularly in the face. They feed on sebum and keratinocyte debris. Infestation increases with age. And recently, D. musculi, one of the strains, Demodex musculi, that infects mice, has been shown to outgrow in SPF colonies. Originally shown by Finkelman and Urban and Bill Gauss in a colony of STAT6 mice, but highlighted from the Rockefeller Cornell Weil MSK group in a number of colonies, and from Yale and the IL-13 knockout colony, that these were in knockout colonies became problematic, but that they were being found in strains imported from other academic communities. And I just want to end with saying that patients treated with dupilumab, anti-IL-4 receptor alpha, the most common side effect, up to 40%, is blepharitis, and 6% is rosacea. And a number of these are associated with increased Demodex density. Now, as the Demodex, it's not really clear. I want to just show you what happens to the wild-type mice. They stay wild-type if they're co-housed, but they clearly get infected. This little dot shows an infected follicle, but it's clearly restrained by the 413 system. The knockout mice have almost every follicle is engaged. The skin ILC2s are activated, and clearly they're infected, as shown by chitin synthase gene in these mice. And these mice are completely reversible by drugs that really don't have antibacterial activity. This was a regimen developed at the Rockefeller MSK group that topically attacks the parasite nervous system so they get paralyzed and expelled. So I just want to finish with some of the things I've said. These tissues seem to contain a range of precursors and mature cells and activated cells that have varying rates of turnover from immature precursors that recapitulate normal development. Upstream perturbations can be quite tissue dependent, and these are amplified locally by eicosanoids and neuropeptides, both positively and negatively. Perturbation can drive differentiation of mature effector subtypes associated with RO gamma T and various X-ILC2s, ILC3 phenotypes. This results in entry of these cells into blood and systemization of this response, possibly to clear space, possibly to systematize the response. This also happens in humans, where you can identify sites of perturbation really by the type of ILC2s that are in the blood. This can impact just distant long-term tissues, if I had showed you, and we seem to be finding this circuit particularly effective in sustaining tissue resilience to chronic eukaryotic commensal and pathosymbionts. And I mentioned all the great colleagues who've helped us, including Andrew Schroeder with bioinformatics, great colleagues in the Saber Center at UCSF. I mentioned the work with the Vel and Gev Labs on the skin ILC2s, some recent postdocs and students who are all just doing terrifically, and collaborators at UCSF and funding. This allowed us to do all this work. So thank you, and apologize for running a minute or two over.
Oh, thank you so much, Rich. That was wonderful. Thanks for sharing those very important discoveries. And also, I very much appreciate that along the way, you remind us that this took much of a global effort of scientists everywhere to really understand the biology of this very important cell. So I appreciate that too. Thank you.
Yeah, I echo that. It was a fantastic talk, Rich. Thank you so much. A lot to think about. And the good thing is that we can listen to it again if we miss something. So yeah. Questions for Rich, as always, will be via Twitter. And so please search for the account Global IMMUNOTALKs, then find the tweet that says ask questions for Dr. Richard Locksley here, and reply to that tweet with your question and mentioning #GlobalImmuno. And Rich will use the account ImmunoSpeaker to answer the questions. So thank you so much, Rich, again, for sharing your knowledge with us. And I hope to see everyone again joining next Wednesday for Dr. Federica Sarlusto's Global IMMUNOTALK, the last one of the year.
Yeah. Thanks, everybody. See you
Happy holidays.
Happy holidays.
Yes.
Thank you, Rich.
Great. Thank you.